we make a laser out of silk, which is a laser that is biocompatible. So a laser you could eat, you could inject in your bloodstream, very tiny little particles that emit laser light. And we hope that one day this will be the base for new sensors, which will tell you that if you have a certain sickness or if you have a certain product in a liquid by looking at the lasing emission and not just the fluorescence.
So we do the random lasing by using a very particular structure. And for that, we have polystyrene beads that are deposited after evaporation of the solvent. And these spheres are firstly covered with silk. Silk is a very white material, so it scatters a lot of light. If you now, inside the silk, you put some molecules, you have a medium that not only scatters light, but the molecules absorb this light and emit more light. And if you are able to do this, like in a conventional laser, you are able to amplify the signal at a particular wavelength, and that's what is the laser for. But in this case, it's random. It's not like a normal conventional laser. Actually, we have a picture how it looks like, and we see that these spheres, once we have made a structure, we can remove them by dissolving, specifically dissolving them. And these white shells is the silk, and also the molecules also, but of course we don't see them. And because the silk is white, the light is like scattering everywhere and is trapped inside the structure. The molecules are being excited by this light increase the gain of the whole structure, and that's why the laser can occur inside. So, for example, here we have a piece of this structure. Basically, it's made of spheres that are covered with silk embedded in these molecules. When we excite it with a laser, the light generated from the structure is collected by this blue box. If it's random, it means that it's emitting in all directions, which basically we are collecting inside the box and directed by a fiber. The problem with this is that we cannot access all the information because we are losing the information of the angles, and because we are trying to also study other type of structures. We wanted to do this kind of experiments with the microscope. And with the microscope, we have the advantage that we have connected with a spectrometer. The spectrometer, the other advantage is that we can do Fourier analysis, and for that we just image the back focal plane of the objective, and this gives information of the angles on where the light has been going. So the nice thing of this structure is that it's made of silk, so this is biocompatible, and this could have a very big range of applications. We are still working on making it small enough to be really biocompatible for the human body, let's say, or in general. But so far we are just reaching the 10 micrometer size needed for the lasing to occur. And because we are wanting to explore smaller sizes, that's why we are going, instead of this blue box that I was showing before, we are just going for the microscope, where we can really excite smaller areas and understand more what is going on. So this positioner, what it does is control the position of this fiber. This fiber is not a normal fiber, but it's a pole fiber. What it means is that when you have a normal fiber and you put some heat to it, and then you pull the two extremes, this part in the middle gets narrow, and at the end it breaks. And this narrow part, one of these narrow parts, is what we have here. And the good thing about this is that it's so narrow that light cannot be trapped inside, so it escapes. And when you get this structure, this fiber next to your structure, this light that escapes can couple to the structure, and then you can study how this structure reacts to it.